Good afternoon, everybody. Now we are going to talk about bioethics in the human embryos edition via CRISPR. It's a proposal I have. At the beginning, I thought by myself about the communication theory. Wittgenstein talked a little bit our communication. So he told something like what one say or what people in general say can change. Ways of life and use of language change. So meaning change, but not literally and instantaneously. Sorry. Things shift and evolve, but rarely, if ever, so drastically that we can lose all meaning. So there is no timeless essence, at least for some, and perhaps all concepts, but we still understand one another well enough most of the time. He's talking about communication, about how words have different meaning, and this meaning are evaluating through the time, and we have different meaning for the same word, and we still communicate. So he is talking about words. In DNA, we have also a very huge book. So we have all the letters that will tell how we would see our life, or at least our phenotype, and the relations between molecules and everything. And now we can change it. So even we can change the DNA and we can take this information, we still, I believe, we will still communicate each other ever. So that's why I brought this theory, the communication theory. I think we have a kind of similarity. We can change several genomes, we can edit several genomes, even human genomes, and we would be the human beings. And I also brought a meaning of the theory or the origin of bioethics. And Potter in the 1921 works about what would be bioethics. So he told something like he combines the work of humanities and scientists whose goals are wisdom and knowledge. Knowledge is defined as the knowledge of how to use knowledge for social good. The search for wisdom is a new orientation because the survival of the man is at stake. The ethical values must be tested in terms of the future, and we cannot divorce from biological beings. And since we are talking about CRISPRs, that is very strong biological stuff, I combine these two theories. At the beginning of bioethics, the origin of bioethics and origin of genome approach are almost the same. In 1971, the Time magazine published a paper called Man and Superman, the Promise and Danger of the New Genetics. So they were talking about the enzymes that could change our humanity, and this man in Superman was more related to philosophical theory. So that's the first time the term bioethics or the neologism bioethics appeared in the world, when they published something related to genetics. So again, we have this connection. I will tell at the end because why I'm bringing several theories. I mean, the communication theory, the origin of Bioethics, and this is the magazine, the 1971. And at the same book, Porter said, actions that diminish the change of human survival are immoral and should be judged in terms of available knowledge and monitoring of survival parameters that are chosen by scientists and humanists. We believe that scientists and humanists would work together. So the question is, how can we prepare for this new phase of genetics without compromising our existence? At that time, in 1971, they were concerned about changing DNA using enzymes, changing DNA and mixing DNA from different types of organisms. Now we can change even the gene. We can change all the sequence of DNA, of the information we have with DNA, and construct new information, construct new sequencing. And this Superman, well, I skipped introduction, but this Superman is more related to Uberman, that Nietzsche was thinking about, the man that construct their life and can make better the next generation. So the Uberman would be a better man. So it would expand our own existence. So at that time, the people who wrote the article related to genetics was thinking about this theory of humanity. It was more philosophical than technical. And in the year 2015, the Chinese start to use this new technique, the CRISPR, that is a technique that uses a kind of combination of enzyme and RNA that can cut DNA in a specific way, and sometimes changing, sometimes completing the DNA, and you can do whatever you want to your genome. In this case, they used embryo that were not available. I mean, at the article they described that they had an embryo with one zygote, one oocyte, and two sperms. So for them there would be not a moral question because the embryo was not be used. But we have since 1999 declaration, UNESCO declaration about the genome that avoid using or avoid manipulate embryos and manipulate human genetics. This is some photos about this experiment. This is for the article, and they had success on changing molecule of thalassemia, beta thalassemia. It's a specific disease. People cannot have the oxygen going through all the body. I have thalassemia, so I cannot be vegetarian or vegan because I need once a week at least to eat because of this thalassemia. And after this, 2 years later, there was another work in China too. But in this case, it was changing a gene related to HIV. So it's interesting. I put this because of this question: whether in the world could the first CRISPR baby be born? They are thinking about this thing. So at the year 2016, let's see, they did this changing of this gene. It's a gene related to immune system, and some people have this mutation, CCR5 delta 3-2, that make people be resistant to HIV. So the embryo this time also was inviable, and they put the CRISPR to change the original gene for this mutation, and they had success. In this case, it's very good because in both cases we have a kind of gene therapy that is more related to disease. The concern is more about using this for eugenic purpose. I think it's the main point. So this is the a new gene that they have manipulating embryo. It was related to heart disease, but it was in August. The last day, the August, I found a paper that was checking if it was successful or not, because the guy thought that they had success on changing a gene related to disease, heart disease, but in fact they are not very sure. I put the all figure because of this. We have this small world, I mean this sphere, and the other way we have we can spread through this space. So we have our dimension, and we have this small dimension. It's our sphere, molecular sphere, that you have several interactions, and it's a real world, a particular world. And this world has their own evolution. That's why I put at the beginning the addition that Wittgenstein think about the words. They are changing, they are evaluating, but still we can manage, and we still can communicate with each other. I think the same is going to have to occur to genome. We are going to change something, but still have the same humanity. I put so the second Chinese team. Oh, it's the next one. The doubts raised. It was I found last night. It was 31 August. Doubts raised about the CRISPR editing in the human embryos related to that heart gene. So I'm not going to be very technical here showing CRISPRs, and people will see that I editing. There are a lot of information after I end my presentation, and you can see how it functions, the CRISPR function, and how the first embryo was constructed. There are a lot of pictures and tables and information you can see. But continuing to this discussion, biotechnology presents this condition for manipulation of genome knowledge. Remember, and thinking about order affirmation. And bioethics has the challenge of presenting possible use of biotechnology developed in genetics, knowledge of how to use knowledge of this new technology. So one present the condition, present how to use this new condition. And it is, the question is, not everything that we can technically run, we must run. So now we can virtually change wherever we want with this technique. And even we have several techniques. These techniques we can change the DNA, but we have RNA silence that can silence after transcription. We have a new system that viruses use to avoid all this virus. All of them are kind of immune system for eukaryotes, for prokaryotes, for bacteria. In this case, CRISPR is from bacteria. We have a new one from virus, so we are able to make any change in our genome. But should we do? That's the question. And precaution, I think it would be a good principle to use. At the beginning, 19th years, we have a kind of moratorium after using the first restrictions, enzyme. All the geneticists that was working with this issue was in a meeting in Asilomar. We have already talked about this in the classes, and they decide to have 4 years waiting and studying to see if it would be a good, to use these techniques. And one of the principles that raised at that time was the precaution. To see if it's going to be, that any risk of serious or irreversible change requires the taking of a measure to prevent its occurrence. So, if we have any risk, so we would at the same time prevent any occurrence of these risks. But now we also, I think now it would be better to use this precautionary principle in terms of having precaution of using CRISPRs on embryos to other purpose, not disease or not health. That's my main concern. And the precautionary principle is an important approach to this problem of CRISPRs. Meets the criteria of a potentially serious and unpredictable risk in measure are not implemented to prevent such risk before its actual damage. So we have learned about some guidelines and some control to these new techniques and some classes also. And for me, the moral issue should be this main question I have put before. Well, if not everything can technically be executed, must be effectively executed. That's the main point. So, some values are placed with this possibility of using CRISPR. For example, eradicate disease. So we have values in contradictions, eradicate disease, counterpoint with the value of the intrinsic genome as a patrimony of humanity. It's in the declaration of UNESCO that it would be better not changing this patrimony. So the physician tells us, but it would be better to our population eradicate some disease. I'm not very sure. I have already tell everybody that I believe in variability, so we would have a stock of variability. To me, it sounds better. But anyway, and the value of science and the value of human being. So we have these values competing each other. But in the fiction, I put one note here. The fiction have already think about changing our genome. If somebody had seen the film Gattaca, the movie have shown how could see a person totally manipulate. So it could be designed at the lab, and you would have the normal person that would be inferior, and the person would be designed at the lab. But the guy at the 1992 could ever think about this possibility be real. Now we have this possibility to change and to present a designed version. So it's a long affirmative we have thinking about me and a doctor from Brazil. It would be good for scientists to consider Jasper, it's a philosopher, warns about the inadequate use of technology as an instrument of domination. To perceive Jonah's appeal to humility and consideration of science advances, and to embrace Mohan's call to build a society based on understanding, solidarity, and compassion. Finally, they were aware of the universal urgency of existential conversion that would justice to right of others, especially to be. So, if we put together all this meaning of preserving human being, we would have a better science. And in case of the use of the CRISPR techniques, should be controlled in a way that would be applied in research and not to reproduction itself, as it could be compromised the next generation. Here we can use the principle of responsibility of journalists, but I'm not going to talk more about this. So the value of this species, human life in humanity would overlap with the value of research aesthetic perfection. So I think we should preserve more the value of human and not the value of science. I think this conflict of values, for me, we should decide for human, not for science. But anyway, some would be decided for science. Even in the case of disease, attempting to correct paths of the human genome would not guarantee its success or happiness. If everybody see the movie, you see that the perfect guy was not happy. He was not successful. He was perfect in terms of DNA, in terms of phenotype and whatever, but the life was not corresponded to this perspective. Moreover, the consequence of this change cannot be completely predicted in the future when this individual reproduces. So these techniques not guarantee that in the future you would have it passing to the next generation, and it not guarantee that you would change effectively our genome. So we need more research. So I would conclude this talking about manipulation of DNA or genome, telling that 150 years ago, Mendel proposed the existence of factor that would determine the hereditary characteristics. Today, we want to determine these characteristics. You see the changing. So or better, we determine the factor that each individual will carry on in its genome. So it takes 150 years to we constructed this factor that Mendel could even see, but he could describe and to hypothesize. And returning to the starting point of our reflection, the man defines his own existence from each. But, in my view, there would be no moral support to define its biological portion as and who might compromise its very existence by manipulating embryos with CRISPRs. So, for me, we must therefore, in Aristotelian field, seek the right measure for the use of these techniques without impeding the benefits it may bring on curing disease, while avoiding abuse of the integrity of our own species. So that's what I think about using CRISPRs as a possibility of manipulation. So if you think about Nietzsche, it's great to have a better generation, to have the Uberman, but I think we could do this with precaution, we could do this with responsibility, and avoiding this total change of the integrity of our own species. Thank you.
Thank you. So we have some questions. So why is CRISPR different to traditional genetic engineering?
In traditional engineering, you have enzyme. There are a lot of techniques. So at the beginning, you used enzymes to engineer. So you, for the first beginning, you put the gene, but you have no, you were not sure in which part of the genome it would be inserted. So they evaluate and start to use virus flanking regions that permit you to find the gene and put the gene at this place. But you would have three portions of the DNA. Later, they start to use RNA to silence post-transcription program. I mean, you would silence the RNA. So the same way we would change the phenotype. And now you use this technique with you have a system that you have we call it CRISPR-Cas. That is enzyme that we call it endonucleases because it cuts DNA in the middle of the DNA in the region that has double RNA that has two parts. One part it's called guide that find the DNA you want to change. In one part that have the enzyme function. So they are together, they find the DNA, and sometimes it complete. I put the figure at the presentation after this conclusion. You have these two types. You can change the DNA. You can cut both parts and substitute, or you can cut one part and the other part is going to be synthesized and you would be two parts later completed. And a new version of this the specific gene. So we can manipulate several things. But I don't study at the new technique that use virus. It must be simple in this technique. Including CRISPR, we have 3 types of CRISPR. Some have several parts, Cas2, Cas3, for example, several steps to change. The simplest system is this CRISPR-Cas that you have the guide and just one enzyme to cut the DNA and to have in one step this change. So it's very easy and it's very cheap to do this kind of technique. At the beginning, may I tell you,
Yes.
at the beginning, this CRISPR were found at the 1980s, at the beginning of 1980s. The Japanese found a region, the bacteria that was repeated. There were several repeated regions, and between the repeated region there was some what they call spacer region. Later, after studying this region, for the first beginning they didn't know what does it mean. They just described this a different region, and then they started to study. So they saw that these spacers was part of virus. Every time the virus enters in one bacteria, she copies a part of the virus and incorporates in their own genome using this CRISPR-Cas system. It's amazing. The next time this type of virus invite the bacteria, she can protect. She can. It's a kind of immune system, and all the bacteria have several spaces. Depending on how many infections she have suffered, and it's amazing this thing. So at the years 2010, they decide to make a engineered CRISPR, because at the beginning you have the CRISPR-Cas occurring in some bacteria, then they decide to make it artificially, and then start to use several organisms, and now in humans too. So, okay.
Any questions?
You had talked about when we should do it, when you should use this process. So, or were you talking specifically just for embryos in this case, or like overall?
This time I'm just concerned about embryos, human embryos, because you can use for several things and several purposes. But in this case, it's more philosophical. Should we change the embryos in this way? Because you would change definitively. The person who is going to born will have this new sequence at the beginning. For a while, we are changing genes that doesn't function, for genes that function.
Right.
But I don't know at the future what people can do. People think about several things, so that's my concern about manipulate embryos in a way that we can compromise the integrity of our own species.
So that's where the bioethics part comes in.
Yeah, we can talk a lot of the techniques. Well, great, it's okay. but my concern is more about this. It's kind of a slippery sloping theory, because if you start to do to disease, it's okay, it's acceptable at the moral point, but the second step is changing for other purpose, and we should have more control to these things. I'm pretty sure we would. I cannot control everybody.
Yeah.
But if we could control most of the research and control the using of these techniques, I think it would be better, especially for embryos, because now, until now, they are using not very viable embryos. Always when people ask scientists, no, it's okay because the embryo is not viable. For example, this last guy that changed a gene related to heart disease, he had a lot of embryos that had extra chromosomes, so they were available, so they couldn't be used to raise a people. Should I say raise, but it was available. So they decided to use, I think it was 83 embryos that they could manipulate in this situation.
That's interesting. That's interesting.
Until now, we have all of them with this condition. They are not viable, but they can be viable one day. I don't know. That's my concern. It's a kind of a concern of should we change definitively our species? Should we definitively change our genetics pattern? I don't know. I still concerned about this.
Yeah.
Physicians always say, no, it's okay. You need to save people. It's better to not have a gene that causes disease. I'm not pretty sure. For example, you have a mutation in Africa that can lead to a death. But some people just live because this mutation. Some protozoaries cannot be in the cell because the cell is have a different condition, this mutation. So I'm concerned about embryos, and I'm concerned about to preserve the diversity we have in our species. But who would decide what would be better to change? What would be the model? I don't know. So we can't have eugenic people decide what would be the best composition for all the molecules.
So should we, for example, if we're good Catholics, we may want to clone the Pope, is that right?
I don't know, maybe.
Who gives consent?
As long as you don't clone Trump.
No. Hey, Ku Klux Klan, maybe. Oh my God.
Church. So it should be the Pope who gives the consent to people who want to clone.
Well, the person who wants to be cloned, of course, needs to give consent.
Yeah.
Are they the owner of their genetic identity? But an embryo has no capacity to give consent.
Yes.
So you can't give consent until you're able to understand.
Yeah.
So it should be the parents first.
Or the parents or the person themselves
Yeah.
as an adult.
Person.
But for example, if you love your child and they die when they're 1 year old, can you clone that child to get the child back again? Because they were perfect and you want to clone them.
And if he died because of a specific gene, you can change with CRISPR. in order to have a health.
CRISPR is specific genes, so
Yeah.
it's curing disease. It's easier in
Yeah.
the
I
existing.
mean, that's what
individual.
I would think that it would be, you could easily use it for, like to cure,
Yeah.
like the stem cell thing. You can use stem cells to help, like with some, like Parkinson's and different things like that. So, I mean, I think if you could use it just for that, so you could change those things, so people wouldn't have those genetic mutations, then that would be for the good. But then you never know. I mean, you're changing.
Yeah, yeah. For stem cells, for cell therapy, it's great to use all these
Yeah.
techniques. But for embryo, it's kind of different. You cannot control, you cannot go back because it's the next generation.
Yeah, that's true.
You cannot control. So, two things. But it's stem cell, not since it's a somatic. It's not a germline cell. You can manipulate. There is no more question for me. There is no more question. But for germline, I think that there is a more question. Specifically to your question, more about changing our species, and we cannot control things.
That can be very scary.
I'm still scary. But remember the question, where the baby CRISPR will born? People are always thinking it's a kind of raining, like a space raining. It's a kind of raining to design a new baby with CRISPR technique, I think.
You mentioned earlier that the first part you mentioned about a cure for HIV.
Yeah,
What
it's
was
amazing.
that?
It's amazing. I'm reading because I got it's
Because it's
paper.
with the spread, the very fast spread of HIV. Then that could have been studied some more, so that, or it has been proven already that it can be some cure.
Yeah. We already have this mutation in our population, people that is resistant to HIV.
Oh,
Remember, I put this, the number of the gene is CCR5. There is a mutation, CCR5 delta, which that people who have this mutation is resistant. So they used CRISPR to put this mutation. It's amazing. I love genetics. Love this all techniques. And,
I think I missed something about an explanation of the CRISPR in the first part of your study. Maybe we can look at it first.
these embryos.
The
Let's see
the
CRISPR.
CRISPR.
CRISPR photo. I see it. I put a lot of things. This is the first paper they did using CRISPR-Cas manipulation. So some were incorporated to me. And I don't think I put any picture of. I even found an industry that already sell CRISPR-Cas. I've read covers of this type of thing. I'll put a lot of things, but I'll try to find CRISPR-Cas to show you. I have a book here at the hotel in Ontario. Okay. So they did several steps to see if they were able to cut the DNA and the CRISPR-Cas for function, but it's kind of technical. You see the sequence it's changing. But I'll find a picture of CRISPR-Cas because here we can have an idea what is.
So CRISPR is it a process of?
CRISPR? It's a process. No, it's a system,
A system.
immune system of bacteria. We are just copying this system. They have, in fact, 3 types of system. 1, 2, 3. CRISPR-Cas is the type 2. The first type, they have several steps with CRISPR-Cas 1, 2, and 3. So you would have three molecules. The second type, we have just 1 molecule, Cas. So you have the CRISPR-Cas. It's kind of immune system. Whenever a virus enters in the cell, the first thing the bacteria would do would be cut the DNA. So how can she find this DNA? Using this CRISPR-Cas. Because you have a guide that is a sequence that is complementary to your target, and the Cas that is going to cut this the DNA. Let's see if I. No, I don't know how. We can find to the internet. Can we find?
Yeah, really? That's okay.
It's easy. There are a lot of pictures, and you can even see a video for CRISPR. My concern today was more files of people.
Yeah.
We can see.
Really proper.
See, it's very easy.
It's almost there.
Okay.
I don't know. No. It's whether it's not.
It's not working on a weapon.
It's not working.
Yeah.
Okay, well thank you very Any other questions? Maybe you can continue explanation of CRISPR later. Yeah.
Yeah.
Sure. Thank you.
Thank you, Nilza.
Everybody.
Thank you.
for waiting.
Thank you, most
Until now.
interesting. Thank you.
Thank you.